Hi students, in this prac we are going to consider ways of genetically manipulating the chicken embryo. So as we mentioned previously, one of the advantages of the chicken is that because embryonic development occurs outside the maternal body, it's readily accessible for experimental manipulation. And so one of the techniques that's used here in the chicken model is electroporation. And electroporation involves the transfer of DNA into the embryo across an electric field. And this has some advantages and some limitations. So the advantage of electroporation for gene expression analysis is that it allows a rapid experiment to be conducted, and it's rather inexpensive. One of the limitations is that only certain cell types can tend to be targeted by electroporation. So I'm going to go over the procedure of electroporation now. In the case of electroporation, DNA is firstly injected into an embryo, into the area of interest, if you're interested in limb development, for example, and then an electric current is applied across that tissue that you've just injected the DNA into. DNA being negatively charged will move to the positive electrode. We use different vectors to deliver the DNA into the embryo, and they have pros and cons. Now, one of the typical ways of delivering DNA is through a plasmid vector. So that's shown here. So a plasmid is a small circular piece of DNA that can be propagated, and your gene of interest can be cloned into that plasmid vector, or a short hairpin RNA. This can be injected into the region of the embryo of interest, and then the electric current applied, and the DNA is taken up by the cells because the electric current loosens the pores of the cells. This tends to be transient, and so you can target a small number of cells and then the plasmid is eventually lost over cell division. A better way is to use a plasmid vector carrying the gene of interest, or a short hairpin RNA for knockdown, in what's called a Toll-2 plasmid. This plasmid has these Toll-2 arms on either side of the gene of interest. They will integrate into the genome in the presence of a co-electroporated transposase enzyme. So the transposase enzyme will take this DNA construct out of the plasmid, and via these Toll-2 arms, which have homology to transposable elements in the genome, it can transfer this construct into the genome. And so this allows a permanent integration of your gene of interest, so that the gene is expressed in cells of interest and in all daughter cells subsequently during cell division. Another vector that people use, and it's one that I use in my group, is a vector called ARCAS. Now, ARCAS is a viral vector, and you can clone a gene of interest into ARCAS, or again a short hairpin RNA, and that can be electroporated into tissues. However, there are certain limitations of these two vectors. Firstly, these plasmids here, with electroporation, you can only really target epithelial cells. Electroporation can't penetrate across a basement membrane, so it's really good for targeting genes or knockdown constructs into epithelia. It's very difficult to actually target things like these plasmids into mesenchyme, so you're rather limited in the types of cells that you can hit or you can target. ARCAS virus, in contrast, this gets electroporated into epithelial cells in the same way as these plasmid constructs, but being a virus, it propagates itself inside epithelial cells and spreads to neighbouring cells, which includes mesenchymal cells. So the advantage of this ARCAS vector is that it can target not only epithelia, but also neighbouring mesenchyme. So it integrates into the genome in the same way as this Toll plasmid, and so it spreads vertically to daughter cells, but also horizontally into neighbouring cells. So that's an advantage over the viral vector compared to these Toll-2 plasmid vectors. A disadvantage of this ARCAS vector is that its payload is smaller. So the DNA that you can clone into ARCAS is really limited to about 2.5 kb. So that's one gene or a couple of short hairpin RNAs. Whereas with these Toll-2 and other plasmid vectors, the payload is larger, and you can inject or clone in here genes that are actually larger than 2.5 kb. So these are the two types of approaches that people can use to overexpress or knock down genes in a rapid way in the chicken model.